Let's go through each one of these stages in more detail. And we'll first start off by talking about glycolysis. Glycolysis, again, is the very first stage in cellular respiration. Again, it takes place in the cytoplasm, and it occurs in the presence or the absence of oxygen. It involves enzyme-catalyzed reactions where 6-carbon glucose molecules are converted into 2 3-carbon molecules of pyruvate. Basically, you're taking glucose and splitting it in half. Only 2 ATP molecules are made for each molecule of glucose. Hence, a small amount of ATP is resulting from the process of glycolysis. So how exactly does glycolysis work? Well, first it's going to go ahead and start, number 1, with priming reactions. Glycolysis begins with the addition of energy. 2 high-energy phosphates from 2 molecules of ATP are added to the 6-carbon molecule glucose, producing a 6-carbon molecule with 2 phosphates. Hence, in this first step, we're having to use 2 ATP molecules to start the process. Second, we're going to go ahead and have what are called cleavage reactions. Then the phosphorylated 6-carbon molecule is split in 2, forming 2 3-carbon sugar phosphates. Notice that the 6-carbon sugar diphosphate molecule is literally just split into 2 to form 2 3-carbon sugars with a phosphate attached. Finally, we have our energy harvesting reactions. In a series of reactions, each of the 2 3-carbon sugar phosphates is converted to pyruvate. In the process, an energy-rich hydrogen is harvested as NADH, and 2 ATP molecules are formed for each pyruvate. As you can see, for each 3-carbon sugar with a phosphate attached, we're going to go ahead and liberate electrons in the process that will ultimately be carried to the electron transport chain. And we're also going to go ahead and form 2 ATP for each of these 3-carbon sugars. The end result is going to be 2 molecules of 3-carbon pyruvate. Now, as I mentioned, a small amount of ATP is formed. We have a total of 4 formed, but we had to use 2 in the process. So what we're going to go ahead and have is a net of 2 ATP molecules being created. Next, we have pyruvate oxidation. The oxidative stage of aerobic respiration occurs in the mitochondria. It begins with the conversion of pyruvate into something called acetyl-CoA. Notice that we already had glycolysis occur and we formed pyruvate. Now, once we're in the mitochondrion, we're going to go ahead and liberate CO2 as a byproduct. But more importantly, we're also going to go ahead and liberate electrons, and those electrons will be carried to the electron transport chain by NADH. Something called coenzyme A is going to then be added to the remaining 2 carbons that were from the pyruvate molecule, and that is going to go ahead and result in acetyl-CoA. The acetyl-CoA will then go on to either form ATP or fat, depending on the needs of the individual. Next, we have the Krebs cycle. This also will take place in the mitochondria, and it consists of enzyme-catalyzed oxidation reactions, where acetyl-CoA binds with carbon molecules that then loses electrons, which are passed to the electron transport chain. Only 2 ATP are made for each molecule of glucose. So again, in this particular case, you have a small amount of ATP being created by the Krebs cycle. So how exactly does the Krebs cycle work? Well, number 1, you're going to go ahead and have acetyl-CoA, which came from pyruvate oxidation, enter what is called the Krebs cycle. The Krebs cycle begins when a 2-carbon fragment is transferred from acetyl-CoA to a 4-carbon molecule, which is called the starting material. Notice in the diagram that you have your acetyl-CoA. What's going to happen is that you're going to go ahead and have CoA released, and the 2 carbons from the acetyl-CoA will go ahead and bind with the 4 carbon starting material to produce a 6 carbon molecule. In number 2, the resulting 6 carbon molecule is oxidized, meaning that a hydrogen will be removed to form NADH. And along with that hydrogen, you're going to go ahead and have electrons. And then it will be decarboxylated, where a carbon will be removed to form CO2. Next, the 5 carbon molecule is oxidized and decarboxylated again, and a coupled reaction generates ATP. Notice in this diagram that the 6 carbon molecule is going to go ahead and lose CO2, and it's also going to go ahead and lose a hydrogen that's going to have electrons. These electrons will be carried by NADH to the electron transport chain. You're going to go from a 6 carbon molecule ultimately to the 5 carbon molecule, and after that you're going to again lose more CO2, and you're going to also lose a hydrogen that will have electrons that will bind to NAD, forming NADH, and will be carried to the electron transport chain. During this process, you're going to go ahead and have the production of a small amount of ATP. And then you're going to go ahead and at that point have a 4 carbon molecule. Finally, the resulting 4 carbon molecule is further oxidized, where hydrogens are removed to form FADH2 and NADH. This regenerates the 4 carbon starting material, completing the cycle. If you look at the diagram, you can see that your 4 carbon molecule is going to go ahead and lose more electrons, and that FADH2 and NADH will carry them to the electron transport chain. You're going to then end up with your 4 carbon molecule, or your starting material, that will then go ahead and be reused by more acetyl-CoA after pyruvate oxidation. So the final step of this process is the electron transport chain. Remember that I said that NADH and FADH2 are carrier molecules and that they carry electrons to the electron transport chain so that they could create a lot of energy. NADH and FADH2 are called dinucleotides. They're composed of 2 nucleotides that have been bonded together. And these dinucleotides, again, carry electrons to the electron transport chain. What exactly is the electron transport chain? Well, the electron transport chain is going to go ahead and be a series of membrane-associated electron carrier molecules, or proteins. These are going to basically be proteins found in the inner mitochondrial membrane. If you look at the diagram to the right, you're going to go ahead and see the white membrane. That represents the inner mitochondrial membrane. Below that, you have the very inside of the mitochondrion, or the mitochondrial matrix. And above the membrane, you have the intermembrane space. The intermembrane space, again, is that area between the outer membrane of the mitochondrion and the inner membrane of the mitochondrion. So these proteins that represent the electron transport chain are going to be found in that inner mitochondrial membrane. And these proteins are important because they're going to go ahead and pump protons or hydrogen ions out of the mitochondrial matrix. Their job is to go ahead and pump hydrogen ions from the very inside of the mitochondrion up to the intermembrane space. Well, how exactly does that happen and why is that important? Number one, as I mentioned, NADH and FADH2 are carrying electrons to these proteins found in the membrane. What's going to happen is that these carrier molecules are going to go ahead and release the electrons to these proteins, to the electron transport chain. And it's these electrons that are going to go ahead and power the proteins to allow hydrogen ions to go from the mitochondrial matrix up out into the intermembrane space. When this happens, we're going to go ahead and have reformation of NAD and FADH so that they can again go ahead and carry more electrons to the electron transport chain to generate more energy. And in this process, you also have water being created as a byproduct. Notice in the diagram that you're going to go ahead and have these carrier molecules release their yellow electrons, and the yellow electrons are going to go ahead and power these proteins that are found within the inner mitochondrial membrane to go ahead and release hydrogen ions up into the intermembrane space. Well, why is this important? How does this ultimately go ahead and create energy? The proton pumps lead to an increase in protons in the intermembrane space. And the proton gradient, where you're going to go ahead and have a lot of them accumulating in the intermembrane space, causes the protons to re-enter the matrix through what are called ATP synthase channels. If you go ahead and you look at the diagram to the right, you're going to go ahead and again see the mitochondrial membrane, that inner mitochondrial membrane in white. And below it, you're going to have the mitochondrial matrix, which is the very inside of the mitochondrion. And above that membrane, you have that intermembrane space. The pink protein that is spanning the inner mitochondrial membrane is going to go ahead and represent one of those electron transport proteins. And what you see here is that the electrons are going to go ahead and power the release or removal of hydrogen ions from inside of the mitochondrial matrix up to the intermembrane space. What this means is that we have a large number of hydrogen ions accumulating in that intermembrane space. And what's going to go ahead and happen is that via the process of diffusion, where things move from a higher concentration to a lower concentration, you're going to have these protons, these hydrogen ions, re-enter the mitochondrial matrix. The proton re-entry, or the hydrogen ion re-entry, drives the synthesis of ATP by a process called chemiosmosis. Notice in the diagram that you're going to have these hydrogen ions, indicated in blue, move back down through the purple protein from the intermembrane space into the mitochondrial matrix. This purple protein is called the ATP synthase. And what's going to happen is that basically this synthase is a channel through which these hydrogen ions will move. They're going to go from where they're in a high concentration in the intermembrane space back into the mitochondrial matrix. And when this happens, you have the formation of ATP. The end result is that you're going to have 32 ATP made for each molecule of glucose, specifically from the electron transport chain. Remember that one molecule of glucose is going to go ahead and produce a total of 36 ATP molecules, and 32 of 36 molecules are produced by this process in the electron transport chain and across this inner mitochondrial membrane. Here we have an overview of the electron transport chain and chemiosmosis. If you go ahead and you look at number 1, you have electrons being harvested and carried to the electron transport chain. What we have here is a situation where electrons are going to go ahead and be created from glycolysis and also from pyruvate oxidation. And we're going to go ahead and then come into the inner mitochondrial matrix in the Krebs cycle. And this acetyl-CoA that came from pyruvate oxidation is going to go ahead and release several electrons. Again, remember, NADH and FADH2 are going to carry those yellow electrons to the electron transport chain. In number 2, electrons provide energy to pump protons or hydrogen ions across the membrane. Notice that you have in that inner mitochondrial membrane the pink, purple, and blue electron transport proteins, and that those three proteins are very busy providing an outlet for the hydrogen ions. They're allowing those hydrogen ions to be pumped from the mitochondrial matrix into the intermembrane space. Then in number 3, oxygen is going to join with protons or hydrogen ions and electrons to form water. Remember that these electrons ultimately are going to go ahead and combine with oxygen. Hence, in aerobic respiration, where organisms are exposed to and use oxygen, oxygen is going to be called the final electron acceptor. In other processes that don't use oxygen, you have other final electron acceptors. But in this particular process of aerobic respiration that we use, oxygen will go ahead and take those electrons that just pass through the electron transport chain. In number 4, protons diffuse back down their concentration gradient, driving the synthesis of ATP in chemiosmosis. Notice that you have your ATP synthase, and your ATP synthase is going to go ahead and be that lavender colored protein that is going to go ahead and be found in the inner mitochondrial membrane at the bottom of the screen. You're going to have a lot of hydrogen ions build up in that intermembrane space, and they're going to go ahead and diffuse down their concentration gradient, going from a higher to a lower concentration. And when they re-enter that mitochondrial matrix, you have the formation of 32 ATP, specifically from the electron transport chain in chemiosmosis. Again, remember, a total of 36 ATP molecules are formed from one molecule of glucose.